The objective for these experiments is to observe and compare the amount of carbon dioxide produced by different amount of yeast in the same amount of glucose solutions.
Next is hypothesis. We think that in the test tube which has the most amount of yeast can make the balloon wider than another. Next is variable. First is independent variable, it is the amount of yeast. Second is dependent variable, it is the diameter of balloons. And the last thing is control variable, it is the amount of sugar, type of balloons, temperature, and type of yeast.
The procedure is as follows. First, prepare warm water to 50 milliliters by using the alcohol burner. Second, mix 20 grams of yeast with 200 milliliters of water. Third, dissolve 100 grams of glucose with 50 milliliters of water. Fourth, prepare yeast solution into the test tube by 0, 3, 6, and 10 milliliters respectively, and also add glucose solution by 5 milliliters to our test tube. Fifth, immediately cover our test tube with balloon and left overnight. 6. Check on the result.
And I'm gonna talk about the result. In the first tube, there was no yeast, so the balloon didn't bloat. And for the second tube, we added 3 ml of yeast into the tube, so the balloon bloated just a little bit. And for the next tubes, we added 6 ml and 10 ml of yeast into the tubes, so the balloon bloated accordingly to the amount of yeast.
What went wrong? In the experiment, we saw that every test tube didn't make the balloon wider because of sugar and yeast was small amount, but when we added the leftover yeast with the large of sugar, it made the balloon wide.